Dear students, welcome to my channel. The topic is chromosome banding techniques.
Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment.
Study of chromosome number and structure by staining the dividing cells with certain dyes and examining them under the microscope for cytologic analysis is called chromosomal banding. Most of the cytological analysis performed on dividing cells, that is metaphase cells, and embryo cell of animals are used. However, the development of cell culturing technique has made it possible to study chromosome in other type of cells. For example, human white blood cells can be collected from peripheral blood, separated from the non-dividing red blood cells and put into culture. The WBC then stimulated to divide by chemical treatment, and midway through division a sample of the cells prepared for cytological analysis. The dividing cells on treatment, when treated with a chemical, disables spindle fiber formation. Such mitotically arrested cells are then immersed in a hypotonic solution that causes cell to take up water. On preparation for microscopic examination, such cells are squashed on a microscopic slide such that the chromosomes are spread out in an unfiltered fashions which can be observed by staining. The stain such as fusion reagent acetocarbon stains the chromosomes uniformly, making difficult to distinguish one chromosome from other. Nowadays, cytogenesis use dyes that stain chromosome differentially along the length. The staining of chromosome is known as banding techniques because stains give rise to pattern of bands along with the length of the chromosomes. Let us see the various type of banding techniques and the procedure. Qbanding. Qbanding used quinacrine stain, quinacrine dihydrochloride or quinacrine mustard, and it is the simplest and the first chromosomal banding method. Quinacrine stained chromosomes show a characteristic pattern of bright bands on a darker background. Because quinacrine is a DNA intercalating agent and a fluorescent compound, the band appear only when the chromosomes are exposed to ultraviolet or UV light. Ultraviolet light causes the quinacrine molecules to shine. Therefore, parts of the chromosome intercalated with quinacrine shine brightly, whereas other parts remain dark. This bright dark banding pattern is highly reproducible and is also specific for each chromosome. Thus, with quinacrine banding is used to identify particular chromosomes in a cell and also to determine if a chromosome is structurally abnormal. Quinacrine is an intercalating agent and it intercalates between the base pairs of the DNA helix. Quinacrine has more affinity for DNA sequence containing adenine thymine sequence. Therefore, fluorescence of quinacrine is enhanced along AT rich sequence and appear bright than GC rich sequence. Quinacrine is carcinogenic. Qbanding technique. First, chromosome will be collected, stained with quinacrine mustard, subject to UV light. Banding pattern shows one region rich in AT bases will darkly stain. That region quenches dye and fluorescence situated in heterochromatin region. When the banding pattern region rich in guanine cytosine bases will lightly stain, GC region quenches dye but do not fluorescence situated in euchromatin region. This shows Qbanding of the chromosomes. See some area fluorescence, that is with the adenine thymine region. Not fluorescence is the guanine cytosine rich region. Next is the G-banding. G-banding is the most frequent used technique for chromosome staining in cytogenetics. Giemsa staining is an excellent non-fluorescent staining technique. Giemsa also create a reproducible pattern of band on each chromosome. It is still not clear why chromosomes show band when they are stained with quinacrine or Giemsa. Bright field microscopy is used for visualization. In G-banding technique, Before using Giemsa stain, there is always a pre-treatment step. Usually, proteolytic enzyme trypsin is used for pre-treatment. Therefore, the process is also known as GTG banding, or that means G banding by trypsin with Giemsa. There is an alternative to Giemsa stain and it is right stain. G-banding also produces same banding pattern as Qbanding along the length of chromosome. Giemsa stain has more affinity for DNA sequence rich in adenine thymine content, hence stain dark, while sequence rich in guanine cytosine content stain light. Here you can see the G-bands, lightly stained and darkly stained regions. C-banding. C-banding is called Centromeric heterochromatin staining. In this technique, before using Giemsa stain, the cell is pre-treated with alkali. Therefore, this technique is also known as CBG staining, banding by base with Giemsa. Centromeric heterochromatin and distal part of Y chromosome containing highly repetitive DNA sequence, that is satellite DNA, is used to stain by C-banding technique. After staining, banding is visualized by bright field microscope. Denaturation of chromosome at adenine thymine rich region occurs at faster rate, resulting in loss of DNA from this region, but not from guanine cytosine rich region. The GC rich region is then stained by Giemsa stain, which appears stained, that is R band. G banding is usually preferred over R banding. However, R banding can be used for chromosome identification. In some cases, R banding is a useful complement to G banding because some small light G band can be more easily detected when they are stained by R banding. R banding is also useful for visualization of telomere sequence at the ends of chromosomes. Telomeres stain dark with R banding while light with G banding. Collect the chromosome. Denaturing with alkali solution, washing with sodium citrate at 60 degrees Celsius for 30 minutes. That is, repetitive DNA renature but unique DNA do not renature. Staining with Giemsa solution, banding pattern at heterochromatin region. C banding of chromosomes. R banding. R banding is called reverse chromosome banding. With this banding technique, the band pattern produced in chromosome is reversed to the band produced by G banding and Q banding. That is, the dark band AT-rich region observed in G banding technique appears light in R banding and vice versa. R banding technique also uses Giemsa stain, but before staining with Giemsa, the slide is heated at 88 degrees Celsius in a buffer solution. Heating causes denaturation of DNA. See, this is the R banding pattern. See here, actually this is used to identify the X chromosome. Dear student, I hope you could understand the banding techniques in chromosomes in detail. Thank you for watching. See you next class with another topic. Don't forget to leave a like and please subscribe my channel.